Yes, we can get started in the interest of time. Good evening, everyone. Thank you for joining in. It's my absolute pleasure to introduce our speaker for today, Dr. Gürkan Yardimci, who is joining us from the CEDAR Center at the Knight Cancer Institute at Oregon Health and Science University. Gürkan's research focuses a lot on trying to understand chromatin organization for healthy and cancerous states. But as the title of his talk suggests, he's also started looking at single-cell data sets for answering very similar questions of comparing healthy cases versus cancer cases. He did his PhD in computational biology from Europe before moving on to do his postdoctoral training at the University of Washington with Bill Noble, and that's how I know him, because we overlapped very briefly during that time, but it was a good time to discuss and potential research ideas and talk about all the interesting things that everybody was doing. And so today he's going to talk about the multimodal characterization of cell states at single cell resolution in the context of cancer.
Yes. Yes.
So please join me in welcoming Gürkan to our center.
Thank you for the kind introduction. Yes. So yeah, since Tamra has nicely explained all of this, I'll just go straight into the talk, right? So in our group, we like building. So our interests are mainly single-cell multiomics nowadays, and I have an interest that I share with Tamra in 3D genome and chromatin organization. So since I joined OHSU, I had a lot of opportunities to collaborate with people that generate lots of single-cell multimodal data, multiomics datasets, right? And these datasets tend to be basically at single-cell resolution, which you guys are all very familiar with, of different modalities of the cell, right? What's going on with the DNA sequence, about the transcriptomics, accessible chromatin landscape. You have the 3D genome, and then you have, things binding to chromatin and the epigenetic states and methylation and such, right? So what we like here, since I'm at a cancer institute, even though I'm actually very much, I don't know if I'm going on record saying this, but I'm just a basic science person, right? So I pretend that I study cancer at my institute. But we just like, developing interpretable algorithms. In a way, this is technology development, right? To make sense out of this data. So, and the algorithms we like to develop, we like to make them interpretable, and we like them to have a very, empirical application in the biology space, right? So the two algorithms. So this is our more or less program in single-cell multiomics, where we try to profile three different kinds of phenomena going on with these cells. So the first one we do is like interpretable modeling of the cell state. The second one I'm not going to be able to talk about at this time. And the third one where we look at is studying the structural variations in the cell at single-cell resolution as well. So that's our program. And before that, I think maybe this is a little bit unnecessary for this audience, but just, to bring everybody to the same page, let's just very briefly do a recap of what we're doing here, right? So we know that the bulk genomics measures modalities in cell populations, right? We have these assays like RNA-seq, ATAC-seq that look at the bulk population transcriptomic state and the accessibility landscape, right? The accessibility landscape generally tells you what are the elements that are regulatory and such, right? So what I'm showing you here is that like a very brief overview of like, okay, this is our cell population. It has different cells at different states going on here, as I coded by the color, right? And then you run it through a box sequencing assay, you get a population measurement where then, we can visualize. And for example, we can see in this population, this gene is being expressed, and there's all kinds of accessible elements around this gene, right? So, okay, this is very cool. This is what we're doing so far. So where do we want to go with this? Now, with the advent of single-cell multiomics, right, as you guys are all very much aware that now we have the ability to do these in single-cell resolution, and now we have the ability to do this for multiple modalities, right? So for example, now we have this, there's been a bunch of these assays coming out, but the commercialized version, the 10x multiome, is a single-cell ATAC and RNA modality, right? So rather than looking at this bulk landscape, now we can look at this data in a different landscape where we have these different cell states here, right? And we have their individual read cells, right? So this gene is on the red cells with a particular peak configuration around it, right? Now we can start linking these elements to different modalities to each other and look at them, model them together within single cell resolution, which enhances our ability to look at cross-modality interactions and basically able to understand what's going on in the cell at much higher resolution. So this is most of the data I'm going to be talking about is more or less this, right? These are actually single cell data. What is single cell data? Tables, lots of cells, lots of features. So the first thing I'm going to talk about is this algorithm that we developed for basically understanding cell states in unsupervised fashion. So we call this the algorithm epiconfig. It's an interpretable multimodal topic model to study cell state. So why is this important, right? So now this data is really, we're seeing that there's a lot of generation of single cell multi-omics data sets. The modality combinations are increasing. RNA attack is the most popular combination, but we have a bunch more accumulating as well. People are doing specific atlases, all the PBMC data sets out there, right? And then they're doing different tissues and such, right? So, and then as we're going to, we're going to start getting to a point where we're going to profile more and more cells and learn more and more about these cells, right? So right now, when we started this project, there was very little competition. And then in the next few years, there was an explosion of methods, right? So for example, the CEDAR integrated analysis, which uses a native nearest neighbors type of representation of cells. People have applied deep neural networks like SCVI and such. There's non-negative matrix factorization-based approaches such as MOFA and such, right? And then there are a few other more interesting approaches like Mowgli, where they also use optimal transport. And this is not the alignment that I'm talking about. I'm simply talking about having that aligned already multimodal data and just clustering it, right? Or like reducing it to a lower dimensional representation and getting a sense of that. What we really value here is that building interpretable models, right? So when we get this clustering, can we also tease out the features? And what can we tease out from them to basically learn what is the underlying clustering telling us, right? So that's what we want to get at. And for that, our solution is to build a topic model. But before that, what I would like to show you is that how we represent the data. And it's pretty simple and intuitive. We're still working off the data itself, which is, so we are looking at the same data, same locus, the same gene, the same two peaks around it, right? So what can you do, right? So this is already a table, right? This is already a matrix. You can do basic dimensional reduction. You can do PCA. You can do different things. They're all straightforward. What we really like to study here as well, that we know the genes, the transcription of an element is closely linked to accessibility around it, the regulatory elements around it. So what we like to add to our model is some sort of cross-modality association. And we have a basic, very intuitive way to represent this, where we try to find coactivity, right? We look at genes that are expressed and we look at peaks that are accessible at the same time. And we also encode that as a binary indicator and we put it in our data as well, right? And we have done different versions of this, but we found that this doesn't lead to explosion of features while keeping things simple and interpretable. So we build this multimodal matrix, right? So one modality, another modality, and cross-modality associations, right? Then what we do is that we take this matrix and we start doing dimensional reduction on this using topic model, right? So what we start is this, multi-omic data here, right? And we built that table that I was just showing you. The cross-modality associations is something we sometimes call hybrid words in our inner lingo. We haven't come up with a cooler name yet, so I'll probably call them hybrid name during the hybrid words during our talk. And we built this vocabulary, right? And I call this a vocabulary because the topic modeling is an NLP technique, natural language processing technique. So we like really calling this a vocabulary. So we build this matrix with different modalities, right? And then we do a dimensional reduction on it, where we learn cells and their representation as a topics, right? And we learn another matrix. So this is the observed data. Then we learn these topics, and the topics are a combination of words, basically. So these are represented by, so basically the modeling works by, these are represented by multinomial distributions, right? There's a multinomial distribution over which topics are representing a cell. Then you have another multinomial distribution telling you that which topics are made of which words, basically, right? And this project is, we collaborate with a postdoc and these two very talented RAs from my group, right? So this is a 3-person effort, Chidam, Nikki, and Aaron. And so okay. And so we learned this decomposition. So how is that useful? And before that, I just wanted to have this slide just give you a little bit more intuition. I'm not going to get into technical details in the interest of time, and topic models are pretty well known at this point, right? But again, the idea is that, in case it wasn't very clear before, so we have this observed data where we have our features as words and our cells as documents. This is the observed data. When you train this model probabilistically, what you get is this decomposition, right? You can think of it as similar to matrix factorizations, where your latent variables or your factors are your topics, basically, right? So we train it. We mainly use a fast implementation of like the standard training of topic model algorithms. It's called WARP LDA. It's pretty fast. And so this is more or less an overview of what we do, right? Okay, so we start with the data, we build this vocabulary, we apply to single cell multiomics data, we largely focus on RNA and attack combination. So to kind of visualize what's going on here is that this is the decomposition that you learn. And what I'm showing you here is the topic per cell matrix, right? So I have these thousands of genes and hundreds of thousands of peaks. I have summarized it into a nice representation where I have these topics corresponding to, this was sorted and clustered by also where we also have the cell type labels that were generated from other Surat-like approaches. So you can see that there's very nice corresponding to topics and cells. So basically we have been able to understand what's going on with which features are, and we interrogate the topics, understand which features are corresponding to which specific cell states, right? And I'm going to talk a little bit more about that. So the nice thing about our algorithm is that, okay, on this lower dimensional representation, we can do clustering, we can do visualizations like UMaps. Obviously, it's a single cell talk, so obviously I'm going to be showing you a bunch of UMaps. So yeah, so we can see that in our lower dimensional space, we very nicely represent, like separate cells from each other, which is satisfactory. So okay, so you can do this with different things, right? So what's interesting about this, or what's valuable about this, is that then we can take these topics, which are genes, peaks, and gene peak combinations, and then we can start interrogating to understand what's going on with these cells, right? So once we identify a topic that's representative of a cell type population here, right? So this is B cell population. There is that topic. Then there are ways you can tease out which are the most representative genes, peaks, and gene peak combinations of that cell type. And you can start doing all kinds of different analysis. So you can do like gene term enrichment analysis on the genes themselves. You can do similar analysis on the peaks themselves. You can interrogate what kind of transcription factors are enriched in these peaks that define certain populations, right? We can start seeing if it's the gene peak associations that we learn are concordant with the 3D structure of the genome. And I'll show you some evidence that is the case, right? So from this data, which has nothing to do with the 3D genome, there's good evidence showing that these peak genes associations correspond to 3D interactions as well. So, okay. So I'll run you through the algorithm quickly, which is, right. Try to demonstrate some of the interesting points here. So first, we'd like to, obviously, once we have this algorithm, we want to make sure it does a good job of clustering these cells. So we applied it to some public 10x multiome datasets out there, right? So for example, we applied to this PBMC data, which is like a landmark data that everybody uses nowadays. So, and then we compare the performance to some of the, well-established competing algorithms, right? So what I'm showing you is that cells put in a UMAP space based on their own latent representations of the cell population, right? And then whether this is just visually estimating or getting a sense of, do we separate the cells? And we color these cells by their cell type labels, right? Which come from the data itself. So you can see that Surat separates them well, MoFo separates them well, we separate them well as well. So this is all and good. Okay, so this is just visual, right? So can we quantify this? So there are different ways to quantify this. We do it by two different ways. One is using the silhouette index, which is basically looking at, for each cell, you look at its distance to its closest neighbors versus the distance to the cells that don't share a label with the cell and the distance between that. So the idea is that if it should be close to cells that are in the latent space that share the same label with it, and if it doesn't share a label with them, it should be distant to it. And if that's the case, silhouette label will give you a high score. So you can see that our algorithm has a pretty high silhouette score, which is satisfying. We originally compared against Surat here, but we realized that due to the ways that Surat does things, it's actually an inappropriate comparison, so we dropped that. But overall, it does well by quantifying the performance as well. If you look at different cell types, there isn't like a very compelling story here, but we looked at different cell types, and indeed some cell types are easier to cluster versus others, and there's an algorithm-based difference. And we looked at purity as well, which is what we ask is that if I look at the cell and look at its k-nearest neighbors, how many of those share a label, right? The idea is that the same cell, a cell should have the same labels with its nearest neighbors. So that's what it measures. And in this setting as well, our model does quite well, right? So this is the multi-omic competitor MOFA. And the other thing we compare against here is this unimodal topic model on just RNA data itself, right? So overall, our clustering performance is good. So this is a PBMC data set, like I said. We applied it to another 10x multi-omic data from cancer cells. This was a mixture of blood cells and cancer cells, right? So I think it was a part of lymphoma data, right? Thank you. So again, our performance is, satisfactory, right? So we see that we are competitive, which is what we want anyways, because we are not really going for a more accurate clustering algorithm. We are going for a more of an interpreter algorithm, right? But yes, so before I go forward, I'd like to also emphasize something, right? So these cell type labels are not orthogonal from the data, right? These also come from the data itself. You look at the data, you look at marker genes and such, or you map them against a reference, and you say, oh, this cell in my data set is closest to this particularly labeled data in this reference atlas data, so I'm just going to call it that, right? So, but given this imperfection, we are doing well, right? So, okay. So I'm going to talk a little bit more about that, but keep that in mind that these labels are orthogonal. So, okay, what is our difference from these 10, 20, whatever methods out there that do a decent job of clustering all these data sets, right? So what we did was that we also wanted to be able to deeply interpret the data and automate this for people that don't know how to do as well. So we have a way, there are basically existing different ways to, once you train a topic model, you can pick the features that are strongly associated with that topic model by looking at that multi-monomial distribution, and then say, okay, these genes, these peaks, and these gene peak associations have it strongly associated with this topic, right? And then start interpreting those features. So for example, here, okay, we train our model, right? Then we built this R-Shiny app, right? And this R-Shiny app basically allows us to automate all of that. Okay, so we can visualize our UMAP and different cell types. Then we can start visualizing individual topics, and we can start seeing which topics are representing specific particular cell types. So this particular topic here represents B cells, right? Then after that, what I can do is that I can do all kinds of enrichment analysis on this, right? So I can do the ontology enrichment to understand what these genes are. I can pick the peaks that are making up this topic, and I can look for motif analysis, right? So I can start understanding which transcription factors are acting on this particular cell state, right? Or at least their binding sites are accessible, right? And then I can start interpreting these. For example, this can get a little bit more bigger than this, but I can start looking at there's encode annotations of candidate cis-regulatory elements, basically a huge data set of enhancer candidates in different tissue types, right? So we can look to see the overlap with these things, right? And then the other thing we can look at is that, okay, when I look at peak gene associations, does it tell me functional associations? So for example, I can go look at 3D genome data. If the peaks and genes are indeed associated with each other, I would expect to see some 3D proximity with them, which I'm going to explain a bit later again. So do I see that as well, right? So this algorithm also allows you to not just cluster the data, but able to get a good sense of what's going on with your data. And it does automate it for you. So we expect people will find this useful. Okay. So we build all of that, right? So, and I was telling you that the labels are imperfect, right? So what we wanted to get at is that then try to understand, and I apologize, this is a bit of a busy figure, but right now just focus on the left side. We wanted to have orthogonal validation of what's going on. So we started studying this, applying this model to this particular trimodal assay, which is called TSeq. So this captures transcriptome for you, attack for you, so it's RNA and attack, but it also captures somewhat similar to CITESeq, the surface markers of the cell, so like the epitopes, right? And this is what we call surface marker abundances here. It's also called ADT, right? So what we have here is this interesting data set where for each cell, we have an orthogonal data that we haven't shown to our model. And what we can do with this data is that then we can start interrogating how do these topics correlate and correspond to surface marker abundances rather than these the labels that you would learn from the data itself otherwise, right? So again, we trained the topic model on this data, just showing it RNA and attack, and then we start looking at the surface marker abundances, right? And I'm sorry, it's a bit of a busy figure. Let me show you this in full sight. This way, it's a little bit easier, presumably. But so what you have here is the surface marker abundances. These are our topics. I think you can visually see that there's a lot of correspondence. We also sorted them by the cell type abundances, right? So you can see that the cell type labels and the ADT markers and the topics have a good correspondence. And this is a PCA reduction of the ADT space for simplicity. However, if you look carefully, you will see that some of these cells that have been labeled a particular cell type by the labeling algorithm, you can see that there is certain ADT, gradients across these cells, right? And the topics actually kind of reflect that here, right? So for example, look, this marker is not entirely seen here, right? And then some of these topics begin to reflect that. So you see that for multiple cell type, for example, here, and in these cells, right? So what does this mean, right? So we have a couple of different hypotheses going on about this. Part of it is that some of these cells might be mislabeled, but also it's possible that some of these cells, since you're just labeling based on our RNA, right? So maybe it's not a perfect labeling way, or maybe there are different cell states your labeling resolution is not capturing. So here's an example of that. But before that, let me show you that so this is correlation between all the markers and the topics. And you can see that there's very strong correlation between specific markers and topics, which does give you extra validation that this is capturing potentially reliable novel cell states if you look at the ADT marker as a orthogonal measurement of a cell state, right? So, okay, so let's just look at one of these examples, right? So these are mate cells, or however you call them properly. I just call them mate cells. So this is a particular type of a blood cell. And so this marker here is their canonical marker, KLRG. So, sorry, TCR-VA72, right? So these are the mate cells here in our UMAP. They have their canonical marker here. But you see a certain gradient of certain markers here, which is corresponding to this topic here, right? So this versus this duality here, right? So these are CD8 and CD4, right? So what we've done a bit of a literature search, and we have seen some evidence suggesting that there are different types of mates that either express CD4 or CD8. And this is potentially something that, this particular labeling is missing. And I think this shows you good evidence that this de novo way of identifying these feature sets that correspond to cell states is potentially a way to better understand and annotate your data, right? I think some of you will appreciate that. I know that a few of the people that I talked to have been working on these problems as well, right? So in the field, very much the standard is to build this algorithm, get some labels, measure the performance, see that, oh, look, our algorithm does well, or our algorithm does as well as this and this and this, right? And then just move forward, right? So I think what we're trying to bring to the table here is that first interpretability and to try to get a bit of a better understanding whether these cells are reliable or not. And I would say, I think I've seen a few other conferences that people have pointed out the weaknesses of these labels as well, right? So, okay, all right. So now, okay, so what is the other thing that the algorithm tries to do? Tries to look at these cross-modality interactions, right? So, okay, so are these cross-modality interactions interesting, right? So we're looking at the same gene again, right? So this is the bulk profiles. So it's being expressed as there's a bunch of peaks around it, right? So what I overlaid here for you is that this is super high resolution high-C data from ENCODE. It's publicly available. It's basically a variation of micro-C, I think. So, so this data tells you, for example, is showing you that if you look at the promoter here, so that when I put my mouse here, this is the epigenome browser. And when you put your mouse here, it will show you the two ends of the matrix. And if you look at this long-range enrichment of 3D contacts, left end is here and the right end is here, right? So you are seeing that there is a peak to gene association here in 3D, right? And what we're doing here is that we try to pick up those associations, right? When the gene and peak is accessible at the same or active at the same time. So what we did then is that we started identifying these gene peak associations, or the so-called hybrid words of different topics, right? And we asked the question, okay, are these picking up 3D interactions that are specific to certain cell types? So here, what I'm showing you here is that I keep finding hybrid words of this nature, right, that are in my topics. I pick a part of that high-C matrix here, corresponding to all the hybrid words here, right? And I keep adding them for a particular topic's hybrid words. The idea is that if this topic is specific to a cell state or a cell type, and when I look at the high-C data from that cell type, I will see enrichment at the center, and it will start disappearing in other different cell types, right? So indeed, this is what we are seeing here. There is some shared, for example, these are different T cells, so there's still some contacts, right? So the enrichment at the center is seen in multiple positions, but you do see that, for example, these loops are more or less disappearing when you go to topics corresponding to different cell types. So topic 12, for example, is a monocell. This is a natural killer cell, right? So you see that the enrichment at the center disappears here. So with some fuzziness or with some noise, we are learning that this 3D interactions, this potential 3D interactions are indeed validated by the data as well, right? So basically, this is similar to the correlative analysis that other people do. There's some stuff from Jason Glenn-Restor that calls it, I think they call this DOR-CSS or such, right? But our model does this for you at the same time as well and picks them up for you at the same time. So yeah, so that's, and then I would like to show a couple of algorithmic things that we did to make these models train a little bit better. So one thing that we did was, so one thing that we noticed here also with these data sets is that, and I think some papers don't point this out, that we found out that some of these data sets are very one modality biased, right? So you can pick this data hoping that it will be multimodal, and you will have some cells that are very heavy on the RNA or very heavy on the attack, right? So for example, this is the lymphoma data set again. When you train on this data, you get this really weird string structure after training the topic model. And what we found out that this is partially because some of these cells are very much attack heavy or RNA heavy, and it tends to pick up topics that are really focusing on one modality, right? And creates this artificial substructure, right? So this is the log ratio of the attack versus RNA. And then, you know, what we decided to do in the interest of time, I'm going to go through this a little bit quickly, but this is prior to filtering this signal, right? And what we found that if we filter these really heavily biased cells using very simple heuristics, we train better models and we start learning models, topics that are evenly composed of different modalities rather than being dominated by one particular modality, which is a desirable property. And I think it's important to actually think about this when you're working with these data sets. Okay, here's the other thing. So the last algorithmic thing we did for us, topic model, is that topic models are expensive to train. So we decided to see if we try to figure out if we can train them a little bit faster. So what we started doing here is that the idea is can we select a representative subset, not an unbiased subset, but a biased subset that keeps the diversity of the cells. So rather than training tens of thousands of cells or hundreds of thousands of cells, can I pick a subset of 1,000 cells, keeping the diversity in there, so can I train my models faster? So the way of doing this, one way of doing this is that you can solve this classical optimization problem, which is the facility location problem, where the classical problem is that if I can place my facilities in a particular city in Paris, why would I put them, right? You wouldn't put them right next to each other, right? You would put them at distances to each other. So you try to optimize a solution where you make sure that the things you pick in your subset aren't close to each other, which is the simple function, right? And then you can greedy solve this algorithm and select the subsets. For example, sorry, this is a little bit hard to see, but this is the full data here in blue. And then when you select 250 cells using this algorithm on this data, and I'm visualizing this on a UMAP here, you can see that it very nicely evenly covers this data, right? Meaning that I keep the diversity in there, remove the redundancy as much as possible, and can train on a smaller data set. So what we did here is that we picked different sizes of subsets, right? And you can see that the training time here improves as you go to smaller subsets naturally, right? While trying to keep the diversity even, right? So the proportions are actually better when you look at a smaller subset, right? Because of the algorithm. So the rare cells, they won't be entirely uniform, but they are better represented here. Whereas on the full data set, some of these things are quite rare, right? So, and then we can train our algorithms with the subset, get the speed up boost on it, right? And then, more or less get the same performance. So full model here on 5,000 cells, right? Which speeds up the training quite a bit. And then you can actually impute your topic from the 5,000 cell to the entire data set and capture the entire thing. As like this single cell RNA-seq, single cell ataxic data sets are scaling hugely right now, right? So it's presumably these algorithms will scale as well. So this will be a useful approach as well. So, okay, so here's our summary. So I'll try to speed up a little bit. So this is an algorithm that accurately clusters cells like the other algorithms. The nice properties of our algorithm is that it allows interpretation of transcriptomic, epigenetic, and cross-modality features. I think I'm showing you some evidence that these are reliable markers of cell states rather than these automatic markers that you're getting, and we can speed up the training of this algorithm. Okay, so here's the second. Can we do that? So the second algorithm I would like to talk about is this algorithm we built for copy number variation, right? So what are copy number variations? So basically these are structural variations of the genome where you have an extra copy of your genome in particular regions, or you have lost it, right? So these can be deletion events. For example, you have lost a part of your genome, or you gain extra copies of it, right? So these are important in particular certain diseases and developmental disorders and cancers, because you will gain extra copies of a gene, misregulation will happen. You might even gain extra copies of chromatin remodelers and such, and then they will give you, they will make your genome not behave as a healthy cell. So it's important to understand this. So here's some evidence, some background on that. For example, in breast cancers, this is studied well, right? So this is breast cancer CMV subgroups. People have shown that different subgroups of breast cancer have different CMV profiles, right? Okay, so how do we do this, right? How do we try to understand this? So this is actually a very nice and intuitive algorithm, right? So, and this is obviously, we didn't come up with this. This was the original way how people have been doing it. Basically, CMVs manifest as outliers in your data. If you have an extra copy, you will have more signal coming from that region. If you have a loss of that region, you will have no signal coming from that region, right? So, but there's also natural variation of the signal along the genome due to certain accessibility artifacts, GC content mappability, and things like that. So you need to normalize for that. So the idea is that get to observe data, try to learn a reliable model where you can predict how much signal you expect to see in the region, then look at the changes, right? Large residual will tell you this is potentially a CMV gain or a loss, right? Okay, so this has been done in bulk, right? The problem with bulk is that this averages over a population. It's cool to do this in single cell, so you can, tease that out easily. But the problem is that single cell data is noisy, sparse, and suffers from dropouts. So how can we? And then the outliers themselves are in your data, right? So you need to make sure you are not fitting the outliers into your model as you're getting the predicted signal. So I'll try to explain to you how we do all of that. So our algorithm is called Riddler. So it's robust in terms of the applications and conditions using linear regression. So it's more or less, it's this intuition here is that signal, simple. You bin your fragments, right? You get the total signal at a given arbitrary resolution. We tend to work with 1 megabase because it helps, but we can go to finer resolutions. Then we try to identify true and technical loss. We basically set a simple assumption that how much signal we expect to see in a given signal, given bin in a single cell based on the total signal. And if it's much lower than that, we decide it's a dropout. So this allows us to try to get a rough estimate of a dropout versus an actual loss, right? And then we do a sliding window correction as well, which allows us to get rid of some of the spurious reads, right? Sorry, before that, we fit a robust regression model, right? So this is how we predict the signal. So I'll explain what robust model does, but here the idea is that we fit a generalized linear model with a Poisson link function, and our covariates in this particular case is the GC content mappability, which is true most genomics assays. And when we apply it to, for example, say ATAC-seq, we use the number of peaks in that region because normally more accessible regions will naturally lead to more signal, right? Then we apply our sliding. Okay, so like I was saying, also your outliers are in the data itself, right? So you must not fit the outliers when getting the expected signal. The way we achieve this is that we use a robust framework. The idea with the robust framework is that it has a different loss function called the Huber loss, right? So the idea is that it's got a piecewise function where you pay less attention to the residuals with larger values, right? And then you can basically figure that out by having this hyperparameter C, right? And we have used the default, and it works pretty well for us actually. So, and then here's the idea, here's the intuition, right? So when you try to do a regular fit to this is the curve that you get, right? If you use a Huber function, this would pay not attention to these outliers, and you would fit something like this, right? In our pretty much the data, the gains and losses manifest this way as well, right? So, okay, so this is a little bit more in detail the same thing, right? So we use a Poisson GLM, right? And then these are for our covariates. The nice thing about this is that if you pick these covariates smartly and adapt it based on the assay, you can make it work on other assays, right? Which is I'm going to show in a bit, right? And the other important thing here is that since it's paying attention to, it's automates not paying attention to the outliers by itself, so you don't need a reference data set. For example, if you do CMV calling on RNA, you need a reference data set to tell you what diploid cell looks like. So you look at the deviations from a diploid cell state. Since we are automating not paying attention to the outliers, we don't need a diploid cell state that is pre-identified here, right? Okay, so this is a visualization of what's going on, right? So this is the normalized reads and the expected values here, right? So our fitted model ratios, right? And here, what I'm trying to demonstrate here is that actually, if we apply the smoothing, we lose a lot of these spurious calls here. This is chromosome 4, and I'm just going to show you evidence in a bit that these are actually spurious losses. So this window smoothing helps as well. Okay, so all right, so first we built this algorithm for ATAC-seq data because there was a lack of a CMV caller for ATAC-seq, right? So here, what we have is that we have this data, ATAC-seq data from SNU 601 cell line, right? So interestingly, even though this is a cell line, this is some clonal structure in it. What I'm showing you here is that, sorry, this is a bit of a busy figure, but 3 different algorithms. These are our competitors here. This is our algorithm. So the bars at the top are ground truth annotations coming from single-cell whole genome sequencing data. There, in this study, they also had paired single-cell whole genome sequencing data. And we get the clones by looking at the CMV structures from the single-cell whole genome sequencing data. There's different CMV signatures in this underlying population. And the heat map under each clone is our CMV codes for each chromosome coming from our algorithm. And you can see that there's very nice concordance and there's very clonal structure R. Oh no. All right, sorry about that. I'll try to talk and do this at the same time. So yeah, the clonality is captured very well, right? And the other interesting thing to note is that, for example, our competitor CopyScat at the time, not a great name, does CMV calling only at ARM resolution. So CopyScat doesn't do a very great job because it's got coarse resolution. So Epi and Viewfinder, which is our other competitor, which does a good job, is missing some CMV events, though, as you can see that it is. So this is our quantitative measurement of the performance, right? So its sensitivity is pretty good, but so its sensitivity is low, right? Because it's conservative. We call a little bit more, we make a little potentially more false discoveries more than Epi and Viewfinder, but we don't miss a lot because they miss a lot, right? So yeah, so in this validation, we saw that it does quite well, right? So then we start applying it to a few other different data sets. So we had breast cancer 10x multiome data from our collaborators. So we took that and we applied it to it, right? So what we did here was that we called the CMVs on the RNA modality using this very widely used algorithm called infer CMV. So for example, here's the MCF7 cells, T47 cells, two different breast cancer cell lines, right? So if you look at the CMVs on the RNA landscape, interestingly, in this multiome data, you don't see much clonality, even though in the other cell line we did, right? If you look at the CMV profiles called from attack, our profiles look, more or less similar to here, right? They are pretty uniform, whereas these algorithms tend to find, due to their various shortcomings, some clonal structure here, which is artificial. What other thing we can do? Another other thing we can do is that, this is not nice and cool stuff like Scott, but we can do something simple here where we say, okay, can I align cells based on their similarity between their CMV profiles and start aligning them accurately, right? If there's heavy CMV signature, I can do a CMV-based integration. So what we did here is that we took these cells and aligned them against each other based on the CMV similarity using the mutual nearest neighbors algorithm. And you can see that when we look at our integration here with Riddler. So the shades of the different colors are RNA and attack modalities, and the bluish ones are the T47Ds, and these are MCF7s. They align pretty nicely, right? But the other two algorithms don't align them all that well. Okay, so all right, it's working very good in attacks, which we're happy with. It's pretty much the state of the attack. In the attack landscape, we have stress tested it. It works quite well. What we did was that can we make this an extensible algorithm? So our collaborators have built technology for doing this on single-cell whole genome sequencing by hacking the 10x ATAC-seq kit, right? So they can do single-cell whole genome sequencing here. And on their data, their existing CMV colors didn't work very well, right? So we started tweaking our algorithm for this. Interestingly, our collaborators' hypothesis, who is Andrew Adey and Konstantin is the lead author in this study. So their idea was that the penetrance of their biochemistry had a gradient across the cell, and it would penetrate better at the periphery and not so well at the nucleus cell, which is pretty much 3D genome compartments, right? So the idea here is that we thought if you look at high-C data, I don't have time to explain it, but it tells you the 3D conformation. But from the high-C data itself, you can infer whether certain bits of chromatin are at the periphery versus the center, right? And then if his hypothesis is true, we had actually, when we looked at the data residuals ourselves, I'm not saying this to brag, but maybe I am, we noticed a very compartmentally looking structure as well, because I've spent 5 years looking at compartments, right? So the idea is that if I take the high-C compartment, which will pretty much capture that where are you in the cell, are you at the periphery versus the center, I can use that as a covariate and normalize for it, right? So we took the high-C compartments and put it as a covariate in our model, and that actually made everything very reasonable. So when you apply this out Riddler with this particular covariate, when you look at this mostly diploid cell, you get mostly diploid codes. Sure, there's some noise, but it's pretty good. When you apply it to this cancer, I think this is a pancreatic cancer cell line, apply the same algorithm to it, you get a lot of CMV events, which makes sense, right? So, okay. So the last bit, right? So this is also something that I think is missing right now is that is there a chemical CMV events on single cell methylation, right? So, okay, so we took Riddler and the intuition being that, okay, if I fix the covariates properly again, I might be able to call it, right? So we took it and we changed our covariates. We dropped the peak covariant because there's no idea of a peak for methylation, right? So, and then we changed our mappability to this basically bisulfite compound with converted genome-specific mappability scores, which is called BizMap from Michael Hoffman's group, right? And then once you do that, we applied our algorithm to this data from old science paper on some colorectal cancer samples. This is one sample where they have different kinds of biopsies, right? So for example, this is the primary tumor, some of this post-treatment, some of this is like lymph node. For example, this is lymph node. Sorry, it's a bit of a busy figure here, but yeah, you can see that we can capture a lot of structure. The thing at the top is the average of the colorectal cancer CMV state from the TCGA, right? So the idea is that if you average our signal here, it would correlate with the average signals, and you do see that. So this is quite satisfactory, right? So, and then this is a competing algorithm. So ours agrees a little bit better with the TCGA. So yeah, and this is an evaluation for performance, but you know, like classically, once you build something, you try to show it's working best, and in this case, it's working quite well. Okay, so with this, we can also try to call the basic estimate of clonal structure, right? So this is very fresh results. Sorry for the rough slide, but basically what we are doing here is that we are doing this very simple optimization of a tree building, right? Starting from the most simple CMVs and start tagging, building a tree from it. So I'm not going to talk about this too much, but this gives you a rough idea of what the clonal evolution is, because single cell allows you to get at the clonal evolution quite easily because it's a single cell resolution. Okay, so how many minutes do I have? I'll try to wrap up in the next 5 minutes, so there's time for questions. So the last thing we did is this collaboration with Hisham Mohammed's group at OHSU, and then this is the person leading the study at postdoc Ryan Malquin, right? So we applied our algorithm to multi-omic data from breast cancer cell types in our in-house data, where we have basically RNA attack measurements of different breast cancer tumors, right? Different types in nasal ductal carcinoma, lobular carcinoma, ductal carcinoma in situ, right? So, and then we profiled it. We also generated bulk data, which is population level, and then we did this consensus CMV calls to be able to get high resolution CMVs, right? So, okay. So what we do here is that we call CMVs on the RNA attack modality with our algorithm. We call CMVs at this is 10x multiome, so the data will look much less nicer than other data sets. So it's much more noisier here, right? But you can see there's good concordance between RNA and attack, right? So let me show you the consensus instead, which is easier to look at. So these are the consensus CMV calls where we harmonize the attack and RNA CMV calls, right? And we get this nice clonal structure. This is a UMAP of the different cells based on the clonal structure. You can see that most of the cells so there's a lot going on here, but this bar here is the cell types, right? And these are largely epithelial cells where you see the CMVs, where we expect to see the CMVs, right? So this allows us to get at the clonality of the data, right? So there are different samples where we see clonality. There are different samples where we don't see much more clonality. But you know, just a quick tease of what we can do with this is that now we can start looking at the clonality and start linking to epigenetics because we have all those measurements all at the same time, right? So for example, here's one sample. These are slightly old figures, so they need to be updated, but more or less the point holds that we can call the CMVs, start clustering them based on the CMV profiles, try to identify different clones, and then look at transcription factors of interest that overlap with CMVs, which is color-coded here, right? The locations of different transcription factors. Then start looking at the transcription factors activity at RNA, attack, and overall accessibility, and start correlating with that clone. And they always don't correlate, right? Because some of them have indirect effects. But there are a few that do correlate with the clonal structure as well, right? So the idea is that we can tease a lot out of this data. For example, if we apply topic modeling to this, which is what we can start correlating, you know, clone-specific topics and such, and get a really nice, clear view of this. Okay, so just a summary, this is led by a very talented postdoc in my group, which is, his name is Travis, right? So Travis built all of this, right? So this algorithm is extensible to different modalities, mainly built for attack initially, but can identify CMVs in different clones, right? Can allow even a basic integration based on the CMV similarity. we extended to different things, and then I'm not showing you this, but we have a preprint up, right? And then we also stress tested it, and it works pretty well for sparser data sets as well. Okay. So, all right, so I'm running out of time, so let me start wrapping up, right? So I just told you about two different algorithms that we've been working on, right? Methods that we've been working on to get a multimodal understanding of single-cell multiomics data. The other things that we're working on that I don't have time to talk right now, and they're not they're not ready yet, but we are also trying to understand transcription factor binding from using single-cell genomics data, right? We would, once that works at the satisfactory level, it will allow us to link all of these events to transcription factor binding as well. And like Tamra was saying, the other interest of my lab is 3D genome, right? So the other things that we're working on is, for example, building this pipeline that we call Hitch, which is a really robust, versatile pipeline for processing of high-C, single-cell, high-C metal, high-C data sets, right? And then we have some projects where we're looking at things like pad heterogeneity and such. So we study the 3D genome folding mechanics, right? And we're interested in building a basic rudimentary integration of high-C and RNA-seq data sets, right? So yeah, those are our main interests, right? So, and I'd like to acknowledge my group here and our collaborators, right? So this picture is from our lab, right? So this is my newborn daughter, which is like 3 months old here, right? So this is my wife, the rest is my group, and we had 2 friends of the lab interloping, so I kind of put smiley faces in their cases to not give undue criticism, appreciation. So we collaborate a lot with the Hisham Mohammed's lab, which is where the 10x multiome data is coming, right? So Max and Brown labs are people that we collaborate with on AML space. They generate lots of single-cell multiomics data as well. AD lab generates lots of interesting technology. So the single-cell whole genome sequencing and the methylation, some of the methylation data sets come from his lab, right? Yeah. So, and then this is our funding sources. So, yeah. Thank you.
And so, questions on the first part. To what extent do you use samples from different individuals, or run on just one sample of it?
We didn't pull from individuals because we don't have different samples from different individuals. So it's entirely possible that there will be batch effects in such, and this might be sensitive to that. But right now, we simply don't have data to do that, at least publicly.
And when you did your cell selection at the facility location problem, to what extent did you consider sparsity or density of expression when choosing cells? Yeah,
that's a good question. So when we do the facility location, so you need a similarity function for that, right? So the similarity functions we use in that space, we don't use the topics because it's prior to the training of the model. So we do some really very quick dimensionality reduction on these, right? So for RNA, I think we do actually a Seurat style simple PCA, right? And for RNA, we do the latent semantic index, right? Which is reasonably fast, right? So, those will be somewhat skewed by the sparsity, right? So yeah, and normally when we do these plots, right, all these UMAPs and stuff, we pretty much check, I didn't show any of that, right? But we normally check the gradients in terms of sparsity. And, you know, like you can see that there's generally a gradient that's driven by sparsity as well, right? So we run a bunch of filters to ensure we get rid of the most deeply sparse cells that are problematic. But yeah, we haven't done a systematic evaluation of that, which is something we can do for sure. But on our rudimentary inspection of it, we don't think that particular structure that we do the subset selection is driven by sparsity.
I have one more quick question, but I won't give up.
Thank you for the talk. I have a question. When you multiply gene by you take into account this?
Yes, that's a great question. Sorry, I forgot to say that. Yeah, so that's a good question, right? Because it can actually explode pretty much, right? So we build different models based on distance. We don't put the distance into the representation, which is something we can do as well, right? Basically, the current strategy that we have is to look at a certain distance away and ignore the rest. There's some studies showing that most of the, how generalizable it is, I don't know, but some papers, early papers from Jay Chandler's lab and other labs, I think they must name from Jay Chandler's lab, that indicated that most of the distal regulation is within 100 kb of a gene, right? So that's kind of what we go by. We focus on 100 kb of a gene, but it's entirely possible for every single stuff. So. That's another technical but interesting question, right? So yeah. So there are lots of different ways to do this, right? We look at perplexity and the delta perplexity. People do very different things. Honestly, in my opinion, it's still an open question not to do that, right? Some people like just choosing it based on this one looks a little bit more better and interpretable than that, right? So we put a lot of effort into it. Unfortunately, there isn't a clear answer coming out of that, right? So because the heuristics don't really do a great job, but I would say that most of the time, as you go from, unless you're at the very edges of that, normally we start from, depends on the data set, but we start as few as 5 to 10 topics and go up to 200 topics. Generally, what we see in this data sets is that the cell type topics tend to occupy. Cells types generally have 1 or 2 topics that correspond to them. And the rest of the uninteresting features, even though we try to filter out for the less variant features, tend to fall into these broad topics like yeah, you can see here, this is not greatest, but here a little bit better here. You can see there are some uninteresting broad topics here like this, right? It's all over the place. There's very big thing. So you just basically end up multiplying these topics. Once in a while, you have some problem with a cell type specific topic broken into very distinct 3 topics. I think that is outlier driven because you can get rid of that by filtering out the outliers from the data. But it is, again, entirely possible that maybe there is some substructure going on, right? And I think that's an open question.
And one more question. Actually, this is for fun parts. Did you do anything different for the X chromosome?
For both parts?
The answer might be different.
Yeah.
It's a question for different reasons.
So for the X chromosome here, you don't really need different. I think we did fit chromosome specific models, and we found that that wasn't really necessary. If it's from like a axon activation perspective, yeah. But what we found in practice is that we can just fit a genome-wide model and it performs pretty well for Riddler. For the topic models, no, we don't really do sex chromosome or specific thing. So, yeah.
We are right on time, so let's conversation.
Thank you. Hope you like.